Now, you know, I'm not saying this is really how it happens with that kind of rapidity or anything, but the point is, introduce some misfolded molecules into a brain where there are lots of normal ones just sitting there, and you will get that kind of effect. More misfolded ones, they in turn go and change or recruit others, and you get the spread of disease. Let me show you a picture of one neuron from the first Canadian cow that was diagnosed with mad cow disease. This is one nerve cell, and you might remember from the picture I showed you a few ago that here's the cell with its extensions going out like that, and over there. This is the center of the cell, the main cell body, there's the nucleus. This brown stuff are aggregations of misfolded prions, prion proteins, misfolded. Look at them, they're everywhere in this cell. And some of these belong to other cells because it's just been a slice through the brain. But I don't think there can be any more graphic demonstration of the absolute destruction that can be caused by these. It's everywhere. Now, this is a fascinating video, and this gives you one more clue as to how that mousetrap effect works. These cows, these two cows, this videotape is from 2005. These two cows appear totally normal, right? And in fact, I'm not a cow person, so I don't know if they do that. But I've been told by the people who take care of them that these cows were in 2005 completely normal, and in fact, because they're part of an experiment, they've been sacrificed each year subsequently, and behaviorally they've been completely normal till now. The amazing thing is, these were genetically engineered to have no prion proteins. So their brains, I was going to sweep these off, and I suddenly realized that might not be a smart thing to do. As no prions anywhere in their brains. Now, if there are no prion proteins on the surfaces of their neurons, then they can't get the disease, because the whole point of the disease is you introduce a misfolded one, it converts, you get more misfolded and more misfolded. But if there's nothing to recruit, then you don't get the disease. So these animals were actually challenged with BSE, prions. They didn't get any disease. But you would think, wouldn't you, that if they'd been genetically engineered to be missing a key protein that sits on the surface of their brain cells, you'd see something wrong with them. And to date, and I was just in touch with one of the guys who does this experiment about a week ago, they've seen a few cells missing from one part of the brain, but that it seems trivial because behaviorally these cows are normal. Now, it would be kind of neat to build up herds of these cows, because they could never get mad cow disease. But it's a beautiful illustration, not just from that practical point of view, that, you know, I don't think the point is so much to build up herds of mad cow resistant cows, but instead to show that, yes, the key part of the disease is you introduce some misfolded proteins, but they've got to have normal ones to recruit, to build it up. Okay. Two more diseases. This deer, this unfortunate deer, has something called chronic wasting disease. This particular prion disease is now, in some senses, the most important. It's the largest amount of biomass of misfolded proteins in the world. Here's the history of this disease. It started in captive deer, or was it elk? Deer or elk in Colorado in 1967. Captive. But soon, wild deer and elk were discovered, and then it spread through the late 90s, and then jumped over to Wisconsin. It's complicated by the fact that it's in wild populations who are migrating along river valleys and getting in contact with other either deer or elk, but also animals are being shipped from one state to another. That's why you likely got the Wisconsin thing, then over in Utah, and then in 2005, it jumped to New York and West Virginia, then Kansas. And so today there are 18 states and 2 provinces in which this prion disease exists. It's mysterious how it's transmitted because they aren't being fed like the cows were. They're not being fed meat and bone meal. These are either captive animals that aren't being fed that, or they're wild animals that have no access to it. But the prions are in the deer's saliva. Maybe if they nuzzle each other, the prions will survive in the soil for years. So maybe when deer bed down, or maybe they go to a salt lick, they could be transmitting it that way. But even though it's been a long time, since 1967, the spread has been pretty dramatic. There are chronic wasting disease animals in both Saskatchewan and Alberta. And I should say, when an entire state is colored, it doesn't mean every deer, every wild deer or captive deer in that state is infected. Here's a bit more accurate idea. This is a tiny chunk of Alberta. That little piece there is represented by this. And each one of these is an individual deer that's been discovered with chronic wasting disease. But here's the issue. It is spreading. It is spreading north. If it spreads far enough north, particularly in Saskatchewan, the range of chronic wasting disease infected deer will intersect the migration routes of caribou. If caribou were to start coming down with chronic wasting disease at any kind of degree of prevalence, it's not huge populations yet, but it's only beginning. That would be a major, major disaster. There are all, huge Aboriginal populations depend on caribou for a lot of things, but for food too. Now, is there any evidence so far? Two key questions. Can deer or elk give this disease to caribou? Experiments are underway to test that. So far, there's been no evidence, but they've been using elk chronic wasting disease prions to try and infect actually reindeer, which are the same species as caribou. But they think maybe deer might be a little bit more infectious, so they don't know that yet. They may not be using the correct strain, or at least they haven't tested all the strains of chronic wasting disease. So at this moment, right now, nobody knows whether chronic wasting disease can infect caribou. Can people get sick from eating this? So far, the answer is no evidence. There was a quite amazing and could have been disastrous event with a sportsman's dinner in New York State, where after everybody filled themselves with venison stew and soup and everything else, they found out that the deer had chronic wasting disease. So those people are all under investigation, or at least continuing investigation right now. It's been 5 years though. But you see, I've mentioned the phrase incubation period several times. And what's fascinating about this is that this process that took a half a second here with the mousetraps actually goes very slowly. And so it can be years. I remember I said there are still people dying of kuru 50 years later. Even one of the huge challenges has been finding experimental animals that will develop the disease in a few months rather than years. So I think those people, my feeling would be those people who attended the sportsman's dinner are going to be okay, but we can't be really sure. So, I want you to really see this difference. Kuru is probably finished as a disease, no more cannibalism, no more cases, except those who were infected before 1960 and have managed to stave off the disease until now. Mad cow disease, there's still the occasional case in Canada and in the UK, but in every case it looks like they haven't entered the human food chain, and there are reasons why they might have had access to infected meat and bone meal. There are also abnormal kinds of BSE that only strike cows later in their life. So that's pretty well done. I want to say a couple of words though about the human version of mad cow called variant Creutzfeldt-Jakob disease. 170 people so far in the UK have died. Every one of them has had a particular genetic constitution, meaning that their prion proteins are a certain shape. And there's another 60 some odd percent of the UK public who have a different genetic constitution, meaning their prion proteins are slightly different, and it doesn't look like any of them have come down with the disease yet. There may be one or two ambiguous cases, but largely it's been one genetic type. So the question is, what about those other genetic types? Are they actually resistant and safe, or are they just taking a little longer to incubate the disease? And while we have 170 fatalities so far, maybe we'll have another 350. Nobody knows. But if you look at the Kuru example, the women, they're all, I think all the most recent deaths are women. They have that other genetic type, not the susceptible one, but the resistant one. And they've managed to stave off Kuru for 50 plus years. So it could be that maybe me, maybe any of you that were in the UK for any significant period of time in the 1980s, you might have mad cow prions in you, but you might be lucky in that your prion proteins are the resistant kind, and maybe you'll incubate the disease for 80 years, in which case you don't have to worry because you die of something else first. That's a happy thought, isn't it? Okay, I want to mention one more disease. And this is not a prion disease, but it's a hugely important disease, and it does have, suspicious isn't the right word, it has tantalizing similarities to the prion diseases. And Neil Cashman's going to get up in a couple of minutes and address that more fully. I'm talking about Alzheimer's disease. This is just a speeded up series of animations just to remind you, if you indeed need reminding, that Alzheimer's disease is an inexorable, devastating disease. As the colors change here, the gray matter, the living neurons in the brain are dying. And if that isn't a persuasive animation, of course this takes place over years. Here's another view, whereas the bluer and more purple the brain here gets, the less gray matter it has. So no one would question the fact that Alzheimer's is a devastating disease. Now, one of the hallmarks of the disease is, again, if you look under brain sections in the microscope, as I showed you earlier, you see something that's kind of reminiscent with Kuru brains and also variant Creutzfeldt-Jakob disease brains, and that's these plaques. They're actually aggregates of misfolded proteins. Now they're not prion proteins, but they still are collections of misfolded proteins, just as the prion diseases tend to induce collections, aggregates of misfolded proteins. And there was a very intriguing recent experiment where they have experimental mice that are quite susceptible to having these, the material's called amyloid, having deposits of amyloid develop in their brains. They took extracts from these mice from their brains, injected them just into the abdominal cavity of other mice, and after a few weeks, they started to see exactly this, plaques, deposits in those mouses' brains. So somehow, these misfolded proteins, not prion proteins, but misfolded proteins, had made their way from the gut of the recipient mice up to their brains and were clearly somehow multiplying. Now, whether it's a knock-on kind of effect like I demonstrated or not, nobody's really sure. But the growth of these plaques looks something like this. Can we